Hello everyone, my name is Dr. Devendra Pratap Singh, Managing Director and CEO of Quench Life Sciences Private Limited. I have done my PhD in organic synthesis from UDCT in 2005 and then worked in different pharmaceutical industries for almost like 20 years. In 2022, I along with my co-founders have formed Quench Life Sciences Private Limited in Cherlapalli, Hyderabad. And our mission of forming this Quench Life Sciences is to give a cost-effective and innovative solutions in the pharmaceutical industry for the drug development of an API. Our proprietary pegylation technology of the drug is to conjugate the polyethylene glycol high molecular weight to the API to form a slow release API which increases the half-life as well as reduces the toxicity of the drug. Our mission is to become a globally trusted partner for the various innovative pharmaceutical companies to provide them a innovative solution and then at the end helping the patients to improve their life. With this, I will take you to our chemistry laboratory as well as the analytical laboratory to show you that how we are performing the research into our lab. So this is one of our equipment called as Radley carousel, and here we develop the proof of concept chemistry where the reaction is done on a very small scale, to develop and generate compound on a milligram scale very rapidly and quickly. Once this reaction is done on this particular reaction, and then in case we need to generate a higher quantity of the material, then we go for a little bigger reactor, which I will show to you in my next few minutes. Once the proof of concept chemistry is being done, then the next part is to generate the material on a little larger scale so that we can use that material for several studies, PK, PD studies. So this is one of the reactor, jacketed reactor, one example I am showing here. We are using for that development purposes. This is 1 liter jacketed reactor and the reaction is happening here. This jacketed reactor is connected to a chiller, and this chiller has the capacity to go from -20 degrees to all the way up to 180 degrees Celsius. As I said that during the proof of concept, we generate a lot of materials on a very small scale, and the next part of the work is that we needed to make sure that we get the right quality of the material after the purification. So I will now show you that how we do the purification of those materials in using our high-tech equipment. Is done into the organic chemistry laboratory. The next part is to make sure that we have a high quality material. So we do the purification using this automated equipment called as Combi Flash. Here the purification is done automatically. We collect the high pure sample and then do the workup and perform the quality check using the gas chromatography system. So in this system, the material which we have done the purification and isolated, we do the quality check. Once the quality check is done, then this material is ready for the PK and PD studies. Quality control laboratory where we check the quality of the material using the equipment called high performance liquid chromatography. So as you can see that we have several high performance liquid chromatography, and the purpose of this equipment is to make sure that the material is what generated into the laboratory is of the right quality. And once we complete and test the quality of the material, then this material we subject it to the PK and PD studies to find out the lead candidate. Once we have the lead candidate identified, the next part is to do the development so that we can supply the material for the clinical trials. And eventually after the clinical trial, the material will go into the commercial manufacturing and production. So this is the kind of innovative work which we do into our laboratory to help our innovative partners to bring the drug into market as quickly as possible so that it helps our patients.